Hello class, my name is Eddie Park or Edmund Park and I'm from Presentation Group 3 and I will be going over the CRISPR-Cas9 gene editing tool. So what exactly is the CRISPR-Cas9? Well, simply put, it's a mechanism of gene editing by severance of a specific segment of DNA, and I'll be going over that more in depth later on in the slide. Moving on, the function of gene editing of this specific gene editing mechanism is to delete, insert, or change DNA nucleotide sequences. And it is believed to be a faster, cheaper, more accurate form of gene editing than its predecessors. So I'll be talking about the origins of CRISPR-Cas9. So the name CRISPR-Cas9 can actually be dissected into two different parts. I'll be going over the CRISPR part first. CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. What CRISPR basically is it's a group of shortened segments of repetitive DNA sequences. And CRISPR is actually a naturally occurring mechanism in bacteria, meaning that bacteria uses this mechanism as a defense method against viral infections. So the CRISPR mechanism originates from bacterial immune system. Moving on to the Cas part, Cas alone without 9 stands for CRISPR-associated proteins. And there's actually a difference between Cas proteins and Cas9. So there's two different Cas proteins. So I'll be going over Cas9 first, and then I'm comparing the differences between the two in the next slide. But anyways, Cas9 is actually a specialized form of protein. It's a nuclease, which is an enzyme that can cut DNA. So the Cas9 is actually like the main character or the talisman, whatever you want to call it, for the CRISPR-Cas9 mechanism. And the Cas9 consists of two RNAs, one of them being a guide RNA. I will also be going over that and why it's important later on. So the comparison of Cas protein versus Cas9 protein. The Cas protein, its function is to dissect a portion of the viral DNA. So once the bacteria is infected with a prophage, the Cas protein will then go over to the prophage, dissect a part of it, and it will incorporate it into the CRISPR region of the bacteria. So what this action accomplishes is the cell remembers the viral DNA and its makeup. So that way it will be prepared for the next time it is infected by the same viral species. Then what happens as a result of this is also the viral DNA is copied into shortened RNA pieces. And that is the entirety of the function of the Cas protein. And what the Cas9 does is it takes these shortened RNA pieces and it binds itself to the Cas9 protein. So Cas protein basically provides the picture of a thief. And then the Cas9 is basically a cop, if you think about it. It picks up the picture, and then it is able to determine who is the thief and who to arrest. So that's that's my poor analogy of it, but yeah. So the Cas9 complex with the RNA copy patrols the cell to detect viral infection, and if the same virus were to infect the cell again, it's the same species of virus. If it were to infect the cell again, the Cas9 complex with the RNA copy of the virus destroys the new viral infection. So the next slides I will be going over it with a visual illustration. So here we can see a phage about to infect the bacteria cell. And once it does, it injects its prophage into it. So what the Cas9 does is it recognizes it as a foreign object. And then what it does is it goes over to it, takes a segment of it, and eventually incorporates it into the group of the bacteria's DNA segments, or the CRISPR regions. From then on, the incorporated portion will create copies of the viral DNA. It will actually make shortened RNA copies, from then which the Cas9, or the police officer in this analogy, picks it up, analyzes it, incorporates it into itself, as shown here. It's part of it now. They're one big unified unit. Then it just patrols around waiting for the infection from the same phage, the same species of phage, to infect the host again. So what the Cas9 can do is recognize it, and they will eventually do what Cas proteins do, which is sever the DNA, which makes the viral DNA inoperable. So yes, there's my poor analogy with poor illustrations. Moving on. Scientists figured out how to control CRISPR-Cas9 as a means of genetic editing in 2012. Scientists found out that CRISPR mechanism was not only bound to viral DNA modifications, and in fact, they deemed it almost applicable to every DNA of almost any organism. So from then on, scientists were able to base the gene editing mechanism of CRISPR-Cas9 as we know today based off of the bacterial defense system. And what they notice is that if they are able to control the gRNA, which the Cas9 protein associates itself with, then they were basically able to control what part of the genome it edits. So given that, scientists bind an artificial custom guide RNA to the Cas9 protein, and the Cas9 protein is directed to the desired gene segment, due to the artificial gRNA. And then, the operation of this is very similar to the defense mechanism of the bacteria, where upon arrival, the Cas9 with the artificial gRNA severs the desired portion of the DNA. What this does is that the host cell actually attempts to repair the severed segment of DNA. This doesn't happen with the defense mechanism because the host cell is actually stopping the viral DNA. So they don't really need to repair anything in the defense mechanism. But for the genetic modification, the host cell attempts to repair the severed segment of DNA because it's artificially, persuaded in a way. So the repair of the cell's automatic repairing of the severed segment of DNA is actually very error prone and leads to mutations that can disable the gene. So in order to counteract this, what scientists do is they add an artificial healthy desired gene sequence. And what that does is it will occupy the severed regions of the DNA, and the new genes are paired respective to their properties, meaning that, A goes with T, G goes with C, all that stuff. And all in all, the result of this is that recombination and replacement of the original gene sequence has occurred once this process is completed. By utilizing the mechanism of CRISPR-Cas9 with the addition of artificial guidance means newfound treatments for DNA error. Diseases and illnesses that are linked to faulty DNA sequences are now likely to be treated. This includes cystic fibrosis, sickle cell anemia, Huntington's disease, and more. And the usage of CRISPR-Cas9 could also yield to improved production of food sources. And today we see them in genetically modified organisms like corn and other food products, as well as the prevention of zoonotic diseases such as malaria and sabotaging actually drug-resistant cancer cells so that drugs are effective against them. That concludes my presentation on the CRISPR-Cas9 gene editing, and this is my reference, and thank you all very much for listening.